alternative pathway of complement system. The alternative pathway of complement system is antibody-independent pathway, as here no any antigen-antibody complex is needed to drive the complement cascade. In the alternative pathway of complement system we need factor B, factor D, and properdin protein. First of all, the C3 molecule in plasma gets hydrolyzed via spontaneous hydrolysis into C3. Along with that, the factor B is cleaved into two fragments BA and BB by factor D. After that, the larger active fragment of factor B, which is BB, combines with C3 forming fluid phase C3 convertase and is actually C3BB complex. The fluid phase C3 convertase works on few molecules of C3 and convert them into C3 on C3b. After that, C3b along with BB combines and forms stable state C3 convertase in the form of C3BBB. This C3 convertase works on C3 proteins, cleaving them and making them active while cleaving them into C3a and C3b. Then along with fluid phase C3 convertase another molecule of C3b and a properdin protein combines to form a complex C3BBBP-C3b. This complex plays the role of C5 convertase. Actually, this complex is analog to C5 convertase of classical pathway. The C5 convertase works on C5 protein, cleaving it into C5 and C5b. On the pathogen membrane, the C5b molecule recruits C6 and C7 and gets into membrane of pathogen. So we get C5b-C6-C7 complex. Then this complex recruits C8 protein and we get complex in the form of C5b678. The complex has capability to polymerize C9 molecule in pathogen membrane. As C5b678 initiates the polymerization of C9 protein to membrane of pathogen, we get poly C9 in the pathogen membrane, which forms pores in the pathogen membrane and is called MAC. The MAC leads to calcium ions concentration increasing in pathogen, which eventually bursts the pathogen via osmotic lysis.